Hi everyone, what I want to do today is review cell structure and cell membrane. This is just a really basic review of vocab terms and parts of the cell, but I think it is important for us to still go over so that you have that foundation for the AP exam. So one of the most important topics when we talk about cells and cell parts is actually cell size. So most of this review that I'm going to go over today is something that we reviewed a lot right before your midterm exam. So hopefully it will all this is just kind of a refresher for you. So cells are really, really small, and that's so they can maintain a large surface area to volume ratio. And what that will do is it increases the efficiency and it increases the rate of chemical exchange. And so if you look at, if you were given an FRQ and you're looking at cell sizes, what you would probably be asked is which one's the most efficient. And you're going to pick the one that's the smallest cell but has the largest surface area to volume ratio. I think students get really confused on that small and large thing. Smallest cell, but has the largest surface area to volume ratio. So that's something that's really important to remember from this slide right here. All right. The second thing on your handout that you printed out is to study the organelles from Quizlet. So instead of you writing down all of the organelles and their functions, I'm just going to breeze through them really quickly, but you have a Quizlet that would really be a good thing for you to go through. And not only is it the organelles, but it's also vocab for the rest of this review that we're doing, cell transport, cell membrane stuff. So that's definitely something that's worth your while to study. I'm just going to run through the different organelles and point out maybe a couple of things that are important. I think everyone knows what the nucleus is at this point. I think what's most important about the nucleus that students have a hard time remembering is that there's actual pores, like little holes in the nucleus that things can go in and out. So remember that DNA is too large to leave the nuclear pores, but RNA isn't. And so RNA can go out, DNA stays in. The other thing that's made inside the nucleus are the ribosomes or the ribosomal subunits. So those are made in the nucleolus, which is the very center of the nucleus. So they can leave also. But I think, yeah, that's just really the most important thing to know is that there's little holes in the nucleus where RNA can leave and ribosomes can leave, but DNA stays inside. And there we have a picture right here. What I would say is important from this picture is that DNA in eukaryotes, remember in eukaryotes, DNA is wrapped around these proteins called histones, and that's the way that it's packaged up and condensed up so that things are just a lot more organized because we have a lot more DNA than prokaryotes. All right, ribosomes are the site of protein synthesis. And then you have free and then you have bound. So your free ribosomes are going to produce proteins for that cell. Your bound ribosomes are going to make proteins for export. All right, your endoplasmic reticulum. I would say the most important thing, I mean, there's a lot that it does, the rough ER and the smooth ER. The rough ER has ribosomes on it, so obviously it's going to be involved in the proteins and the packaging of the proteins. Makes replacement membrane. Smooth ER synthesizes lipids, metabolizes carbs, detoxes drugs and poisons. I would say for the purposes of the AP exam, you really would only need to know this. The rough ER is for packaging proteins. And it's easy to remember that because rough ER has ribosomes on them. So that's what I would probably know from that. You don't really need to know anything from this picture. Right, the Golgi apparatus. The only thing you need to know about the Golgi apparatus is that it synthesizes and packages materials for transport. That's really all you need to know from this slide right here. Right? Lysosomes are really important because they have hydrolytic enzymes. So remember, lysosomes are made in the Golgi apparatus. They're produced by the Golgi apparatus. So they're part of that endomembrane system. But they contain hydrolytic enzymes that help with programmed cell death. So hydrolytic enzymes, remember, are going to be involved in hydrolysis, which are going to just break things up into their little subunits or little pieces, basically. right? Vacuoles are just large, they're membrane bound. They're basically membrane bound vesicles, but they're large and they really just stay in one place and they store things. So remember, plants have that large central vacuole that's going to fill up with water. And that's what's going to help a plant become turgid, is that it fills up with water and then it pushes on the cell wall right there. All right, the mitochondria, we do not say the mitochondria is the powerhouse of the cell. We do say it is the site of cellular respiration. It is the site of ATP synthesis. All right, and then you see these two funny words, cristae and matrix. Matrix is the very center right here, and cristae are these like folded inner membrane, the folded inner membrane. All right, so why is it significant to have the cristae that's folded like this? Well, it increases surface area to volume ratio is what it does, and it makes things more efficient in a more compact space. So that's one thing that's important. The other thing, remember, that's important about the mitochondria is that it has its own DNA and it has its own ribosomes, and it is self-replicating and it is double membraned. And so what we said about that was, that leads us to believe that the mitochondria was once a prokaryote-like thing that was engulfed by a eukaryote-like thing, and then instead of actually eating it and digesting it, the eukaryote, they formed a symbiotic relationship. So the eukaryote ancestor then became a eukaryote. So that's what's important about the mitochondria. Same thing with the chloroplast. It's double membrane, has its own ribosomes, has its own DNA. It's self-replicating. And so it's believed that it was once its own prokaryote-like thing that was engulfed by a eukaryote-like thing. Okay, site of photosynthesis. That's really it for chloroplast. All right. I don't think I've ever seen a question on an AP exam about peroxisomes. I wouldn't really worry about peroxisomes, except I think it's interesting peroxisomes are involved in the production of hydrogen peroxide as a byproduct. So when a peroxisome breaks down alcohol, hydrogen peroxide is the byproduct, which is also toxic itself, but less toxic than alcohol. And so there's an enzyme from the liver that comes in and further breaks down hydrogen peroxide into water and oxygen. And that enzyme is called, do you remember the name of the enzyme that breaks down hydrogen peroxide into water and oxygen? Yes, you are right. It is catalase. I would definitely not worry about intermediate filaments. I would not worry about microfilaments. I would just know microtubules. They're made up of protein tubulin, and they provide a track for organelle movement. I think that's important. But most important about microtubules is they form the spindle for mitosis and meiosis, and then they're the component of cilia and flagella. So I would just know microtubule and basically these two bullet points right here. Centrosomes are the region that microtubules grow. I wouldn't focus too much on memorizing that. If you don't remember that, that's not that big of a deal. Flagella and cilia, remember flagella is just like one little whippy tail that helps things like bacteria and sperm move through water. And then cilia are really short and numerous. So they both do the same thing. It's just when a bacteria or something, a cell has a flagella, it just has one long, maybe a few, and then cilia, there's lots of them and they're short. And just to know that cilia and flagella are made up of microtubules with motor proteins. Right, the extracellular matrix is like this barbed wire looking thing on the outside of the cell, and its job is to strengthen tissue and transmit external signals. So it looks like the barbed wire thing, made up of glycoproteins. Glyco meaning sugar, protein meaning protein. So proteins with sugars attached to them. So just extracellular matrix on the outside, strengthens, helps transmit signals. I've actually never seen any of these words on the AP exam, but just for completion's sake, I did put them on here. These are just ways that two animal cells can be joined together. So they can be joined in a tight junction, which is like a watertight seal, like your skin. desmosomes that are like rivets that fasten the cell in strong sheets like muscle tissue, and then gap junctions, which are like the holes or channels through which ion sugars and small molecules can pass. So your heart, the cells of your heart are going to have a lot of gap junctions. But I have never seen those on an AP exam, but I just put them on here just for completion's sake to let you know that, hey, cells are joined together in a few different ways. But then if you don't memorize them, I don't think it's that big of a deal. Same thing with this one. It's definitely important to know that the plant cell wall is going to protect it and maintain its shape, and it's made out of cellulose. Plasmodesmata are like gap junctions. So you see the gap junction for the animal cell. Plasmodesmata would be the thing that's just like a gap junction, and plant cells only have plasmodesmata because of the cell wall. So just so you know, hey, plant cells, animal cells, they're joined together. They can actually send things to each other through these junctions. All right, on your handout, I think I've given plant versus animal cells. That was the next thing to write down. So if you want, what I would do is I would pause the video right now and I would write these things down because I think this is something important. You know, if you're wanting to have something in front of you on the day you take the AP exam, you can. They're going to give you 2 FRQs, 4 parts each, so 8 questions in 45 minutes. It's going to be really hard to use notes, but I would definitely write this down as something that you kind of use to cram the night before or something like that. So you're going to want to pause right now. All right, and then I'm going to move on. Same thing here, prokaryote versus eukaryote, really, really important. I think everyone at this point knows that prokaryotes have no nucleus, eukaryotes do. Prokaryotes have no organelles other than ribosomes. Eukaryotes have all the special membrane-like organelles. And then prokaryotes are small. These are large. And I'm going to give you a chance to pause in just a second, but I want to add something to this. So prokaryotes are smaller, more primitive. Eukaryotes are larger, more complex. Please be sure to write down, prokaryotes do binary fission. Eukaryotes do mitosis and meiosis. I would definitely write that down. Prokaryotes do binary fission. Eukaryotes do mitosis and meiosis. So if you were asked, do prokaryotes form a spindle? No, because they don't do mitosis or meiosis. There's no reason to form a spindle. The other thing that I think comes up, it was just in my mind and now I totally forgot it. No mitosis, no meiosis. Oh, I remember what it is. It's that prokaryotes have one singular chromosome, circular chromosome, and eukaryotes have multiple linear chromosomes. And then another thing that we've looked at before is that prokaryotes, when they regulate gene expression, they're going to use operons. So remember the irrepressible and inducible operons. So something to look forward to. We'll review that in 2 weeks. And then eukaryotes are going to use that transcription initiation complex and those inducers and stuff like that. So we'll review that in a couple of weeks. So if you need a pause, go ahead and pause to write this down. All right, I think it's kind of important here just to kind of see a path of a protein. So it says, yeah, at the bottom, okay, it says study cell membrane vocab on Quizlet. So here we have the nucleus. This is where DNA gets replicated. This is where RNA is transcribed. RNA then leaves the nucleus through a pore, goes to a ribosome. It could be a bound ribosome if it's getting packaged for secretion, so that's the path we're taking. So once the protein is made, it's packaged in a vesicle from the ER. It is passed to the Golgi, where it is then packaged in and modified a little bit more. It's modified for shipment, basically. And then it's sent to the cell membrane. The vesicle fuses with the cell membrane and expels the contents there. So at the bottom it says, go ahead and study cell membrane vocab on Quizlet. These words are included. Selectively permeable. You guys already know what that means, so I'm just going to skip through this. This is the beautiful picture we see of our cell membrane. We have our extracellular matrix out here. Remember in our pictures, if it's purple, it's a protein. If it's green, it's a carbohydrate. Then we have our lipid bilayer. Our lipid bilayer is made out of phospholipids that have a hydrophilic head and 2 hydrophobic tails. So it's the phosphate and those negative charges that make it hydrophilic. Because remember, charges are attracted to water. And then it's the hydrocarbon, long hydrocarbon tails that make these, or long hydrocarbon chains that make these tails hydrophobic. And so it's a lipid bilayer. It makes it selectively permeable because not anything can get through. So what can get through the cell membrane? Small molecules like water, oxygen, carbon dioxide, and then lipid, non-polar molecules, no matter how large they are, like testosterone, estrogen, stuff like that. All right. So amphipathic, that's in the Quizlet, means that it has both properties. What makes the phospholipid amphipathic is the phosphate head, which makes it hydrophilic, and then the fatty acid tails, which makes it hydrophobic. So that's on your Quizlet. You don't need to write that down. Membrane fluidity, this is the next question. How does the membrane adjust to cold temperatures? So to cold temperatures, you're going to see kinks in the tails, and those kinks are caused by double bonds between carbons, which makes them unsaturated. So what that does is it prevents packing. Cholesterol is also going to resist changes. So at low temperatures, you have those unsaturated hydrocarbons, the double bonds between carbons, which causes a kink in the tail, keeps it from packing too close together and solidifying. Cholesterol is going to also keep them from packing together and causing them to solidify. But the other cool thing about cholesterol is not only is it going to prevent packing during low temperatures, it's going to prevent the cell membrane from becoming too fluid during high temperatures. So cholesterol is super cool that it does both of those things. You know, cholesterol just gets such a bad rap, but it's so important. I heard that when someone, and I'm going to be talking about coronavirus a lot tomorrow especially. I kept myself from talking about it just because I knew people were stressed, but I'm just going to go ahead and put it all out there and just start talking about it because it is so relevant. But I find what's interesting is that they say like 3 or 4 days after you start showing symptoms of coronavirus, cholesterol levels significantly go down, which causes a person to not be able to fight the infection well enough. So I thought that was really interesting that if someone has high cholesterol and they're taking a cholesterol drug and they get coronavirus, I think I've heard that some doctors might actually tell them to either lower their dose of the cholesterol drug or stop taking it completely. I mean, that definitely could backfire, but I just found that to be really interesting. So cholesterol is really important and it is very necessary. Obviously, if you have too much of it, it can be detrimental, but definitely has its place in cell functioning. Integral and peripheral proteins, you just need to know integral are embedded within. Peripheral are on the side. I wouldn't worry too much about everything written on this slide except those two things. All right. And beautiful. Here's your integral. Here's your peripheral. I love that picture. All right, transmembrane proteins. So these are your integral proteins that are going to have a hydrophobic interior and hydrophilic ends. And so it says, why are transmembrane proteins, so transmembrane proteins, they span the entire membrane. Why are they amphipathic? Well, if they weren't amphipathic, they would get pushed out. So they have to span the entire membrane, which means they've got to have hydrophilic properties here, hydrophobic properties here, hydrophilic properties here. So when able to span the entire membrane, they have to mimic or match the membrane properties. So you can pause if you need to. All right, here are the functions of membrane proteins: transport, enzymes, signaling, cell-to-cell recognition, intercellular joining, and attachment to the cytoskeleton. What I want to talk about tomorrow, I'm going to focus on this one right here and how proteins within the membrane function as receptors for cell communication. Super interesting. All right, the carbohydrates, the function of the carbohydrates on the cell membrane are for cell-to-cell recognition. So remember when we did our blood typing and we talked about the different blood types and how if you have someone with an A blood type who gets an infusion of B, those B blood cells that they get in their infusion are going to have these certain carbohydrate chains that A blood type person's body is not going to recognize. Therefore, the immune system is going to attack the blood and form clots, and that person could die. So carbohydrates are very important for giving the cell it's almost like its ID card, basically. Or I would say maybe even like passport, because you can get infusions of someone else's blood, obviously, and it match. So I think maybe it's more like a passport. I don't know. I just did that off the top of my head, so hopefully I didn't mess anyone up. All right, so simple diffusion and osmosis is a form of passive transport, which doesn't really require much energy. So diffusion is going to occur down a concentration gradient. So simple diffusion and osmosis is when no energy is needed. Diffusion is going to go down the concentration gradient from high to low concentration. Sorry if you hear my kids screaming in the background. They're playing with a dog. All right, so I think everyone pretty much remembers this. Okay, and then when we get to simple diffusion, we talk about the simple diffusion of water or osmosis. So remember here, hypotonic means water moves in. Water is going towards solute, which is bad for animal cells, good for plant cells. Isotonic means there's no net movement. Water is always going in and out, but there's no one side doing more than the other. There's not more going in than going out. It's just equal. And so that's good for animal cells. That's not that good for plant cells. They'd be flaccid. They need, and you'd have to water them to like revive them. And then hypertonic is bad for both. That's when there's more solute on the outside, water leaves, the cell shrivels. Okay, I do not anticipate any water potential problems on this year's exam. I don't anticipate you working them out, although they are saying you might need a calculator. They're saying to have a calculator on you. But I don't anticipate a problem, but just so you know, water is going to move from a high to a low water potential. Water moves from where there's less solute to more solute. So as solute goes up, water potential goes down. And we kept talking about this over and over again. It's almost like, counterintuitive. But as solute goes up, this right here goes down. And if you remember, the way to calculate solute potential is negative I CRT. So the more concentrated your solute gets, the more negative water potential gets. So I would just remember water moves from a high to a low water potential. The more solute you have, the lower water potential gets because of that negative sign. I think I got all that on there. I don't want to take up too much time on this thing. Okay, we're getting there. All right. So that's water potential and that's osmosis. Facilitated diffusion. So if you facilitate something, you help, basically. Facilitated diffusion are for those substances that need to get across the cell membrane, but they can't freely pass. So they're going to need a special protein to go through. And examples of things that will need facilitated diffusion are ions, like a sodium ion, potassium ion, chlorine ion, something like that. And then polar molecules like water and glucose. Now, water can freely pass through the cell membrane to a certain extent, but remember, if you need large amounts of water to pass through the membrane, like with the kidneys, you're going to want an aquaporin, which will allow the quicker passing of water. So facilitated diffusion just means there's still no energy that's required. Still no energy required because you're going from a high to a low concentration. That substance, though, couldn't do it by itself. It needed a channel to pass through, basically. All right, and then we get to active transport. Remember, active transport is going to require energy because we're going from a high to a low, going from a low to a high. It's the opposite of what was happening. So it goes against the natural tendency of equilibrium to be established. So proteins are going to go against their concentration gradient. And the two active transport pumps that are really important are the sodium potassium pump and the proton pump. So remember, the sodium potassium pump is going to establish that membrane potential or an electrochemical gradient that is necessary for neurons to fire. So kind of think back where we talked about 3 sodiums out for every 2 potassiums in will lead to the outside of the neuron being much more positively charged than the inside, which creates a membrane potential. So then when a sodium channel opens, when you have an action potential and the sodium channel opens, those sodiums are going to rush into the cell, creating the action potential and the signaling that moves down a neuron. So those are really important. Proton pump, remember, these are going to be involved in cellular respiration and photosynthesis. Hydrogens get pumped against their concentration gradient, which creates a membrane potential, or the capacity for for work or something to happen. And remember, with cellular respiration and photosynthesis, those hydrogens desperately want to get back in to establish equilibrium. They're going to pass through ATP synthase, causing that rotor to turn and for ATP to be formed. So electrogenic pumps are so important. They do require energy for them to occur, though. All right, and then co-transport. I have seen some AP questions on co-transport. You're basically just coupling an electrogenic pump with facilitated diffusion. So the first thing that happens is ATP is used to pump hydrogen against its concentration gradient. For whatever reason, the drive of hydrogen rushing back in is going to help sucrose come in with it. So they go hand in hand, they come in. So this is just a way to get sucrose into the cell that actually costs a little less energy than if the cell were to try to transport sucrose on its own. It's the drive of hydrogens back in, that brings sucrose back in. And I don't think there's really a whole lot else I need to say about this one. There was a question about co-transport a couple of years ago that was really difficult, but the picture really shows you everything that you need. All right, and then this is the last slide. Bulk transport. Bulk transport is going to not use something's not going through the membrane. Something is not using a protein embedded within the membrane. This stuff that's coming in or going out is using the actual membrane itself. And so the membrane, remember, is going to bud in and form a vesicle and the vesicle. So these are for things that are way too big. So this is actually how a virus would get into your cell. This would be a virus is going to be a receptor-mediated endocytosis, where the virus is going to have a protein. I'm going to talk to you all about coronavirus tomorrow and the actual protein that it has on its surface that matches the receptors of some of the human cells. That it's almost like coronavirus has a key, and your receptor is the lock, and it fits that lock so perfect, and it unlocks your cells, and then your cells will bring it in using the membrane. Then when a virus is done assembling, your cell and the machinery of your cell is done reassembling more virus, it's going to expel it back out to infect other cells. There are other things that are involved in endocytosis, exocytosis. It's not just viral infections. Endocytosis is what your macrophages do whenever they're engulfing a pathogen. It's what your cells do whenever they need large bulk macromolecules. It's what your cells do whenever they're releasing proteins that are to be sent to another cell, maybe. So there's a lot of things that this can be used for, but it's bulk transport. Yeah, and then I think what I really think is important here is the receptor-mediated endocytosis. That's going to lead us into our discussion on coronavirus tomorrow. So what are we going to do tomorrow? How can we relate? What topics can we relate to this review? So Wednesdays, remember, our higher level review, not just basic. Wednesdays are going to be when I'm trying to connect ideas across our curriculum. And so I'm going to talk about cell communication, immune system response, and then COVID-19, the coronavirus. I think I was supposed to capitalize all that, whatever. COVID-19, the coronavirus, and how exactly, very detailed, how it infects the cells and what is being done to try to treat it right now. All right, so if you watched this, thank you so much for participating in this review and for trying to get a higher score on your AP exam. I do know a lot of students that told me last year they did not want to review for the AP exam, and then once they got their score, they were so happy that they were able to use it. So I do appreciate those of you that are choosing to watch these videos. All right, I will talk to you guys later.